bring those questions after you've studied a bunch over the weekend. Bring questions Tuesday. Okay, any questions on anything I've covered? I'm going to finish up chapter 9. Any questions on the SOS response? The regulon that lacks A represses a number of different operons, and how Rec A is involved in its autodigestion and then turning on of this whole suite of genes that are involved in DNA repair. Okay. Let's see. So horizontal gene transfer, we've talked about this a couple of times. What is interesting is, we introduced the chapter talking about a chromosome being a mosaic of genes. And so there's some non-uniform genes that are interspersed with uniform genes. And there's these islands of genes that don't look like they originally belonged to that organism. So they're called genomic islands, and the example here on the left is discussing different strains of E. coli. So strain K12 is one of the original laboratory isolates that is harmless. We all harbor E. coli in our gut, and it benefits us. But some strains of E. coli have acquired genes that makes them toxic, makes them release toxins. And so a lot of these O157 strains kill our babies because these toxins overwhelm an infant's immune system and really just the fact of their size. And so these genes have been acquired and are on all islands. These toxicity genes are on groups of genes that are all organized together. And so where is some evidence for this acquisition from the outside, is that all organisms have a characteristic content of guanine and cytosine. And so some organisms have, say, 65% guanine cytosine content, a lot more than half. Some organisms have 45% GC content. And that's true of almost their entire genome. And there's also a couple of other distinctive traits of that organism's and that species' chromosome. So all of a sudden you run into, if you have a 60% GC chromosome, you run into an island of genes that's about 40% GC content. And then it resumes 60% GC content, as is the characteristic of your species. So that interruption of kind of normal guanine cytosine content, and I'm just talking about concentration among the entire chromosome, shows at least evidence for acquisition from another organism. And so those are interrupted, and they're discrete sets of genes, so they're called cassettes or islands of GC, either. So the two examples is islands of genes that are carrying virulence factors that make an organism pathogenic, groups of genes that allow that organism to interact symbiotically with a host. So on the right is not much detail is required here, but there is this ability to insert, and it's interesting, it's often right next to the gene that codes for a transfer RNA. And so right here on the right here, it's shown phenylalanine transfer RNA. It's just as a typical insertion site for these pathogenicity islands or symbiosis islands of genes. So again, this is acquired from organisms in your same generation horizontally, rather than acquired vertically from your mother cell in terms of being a bacterium. So that was our original distinction, horizontal versus vertical gene transfer. really an important method of acquiring new genes. So we've reviewed in this chapter the three mechanisms by which organisms acquire genes horizontally. So conjugation, transformation, and transduction. So those are three mechanisms by which horizontal gene transfer occurs. So make sure you connect that part of the lecture with this. Okay, the last slide here is we've been talking about acquiring genes. There is an interesting evidence of loss of big groups of genes among bacteria. So pathogenic Shigella exhibit chromosomal, quote, black holes, lacking genes that occur in closely related E. coli. So the interpretation is these Shigella species have lost those genes that they had in common with E. coli. It's even acquired. It's even associated with pathogenicity. So one a pathogen, Bordetella pertussis, the causative agent of whooping cough, is actually 100,000 base pairs smaller than the genome of the less pathogenic species. How about Mycobacterium leprae, which causes leprosy, is composed of non-functional genome. And the idea is that these genes are no longer needed now that this species of Mycobacterium lives inside cells, lives inside of our cells. So there's a pretty interesting correlation with these obligate intracellular parasites that only can live in a eukaryotic host and loss of function. Why? Well, they are using the host cell machinery, the host genes, to do a lot of their work. So they no longer have to have those genes, and so they, the interpretation is they've lost genes over time. And there's this interesting correlation with these parasites having small genomes. And so those are all some evidence of actual loss of genes over time, especially associated with pathogenicity. So it can go either way. We just talked about acquiring pathogenicity genes, and then actually the loss of genes is associated with pathogenicity because you're living inside the cell and you are using cellular genes for those functions. Okay, so that is the end of chapter 9, and I sent out chapter 10 yesterday afternoon on Canvas. People get chapter 10 on Canvas? Thanks. So this is all about gene regulation. And you've seen some of this information in Biology 211, so it's a reminder of the lac operon. And then we're going to be looking a little bit at about sigma factor regulation and then finishing up with quorum sensing. Okay, so the idea is you've got thousands, we have thousands of genes. Sometimes we need them, sometimes we don't. So it makes sense to turn them on when we need them and turn them off when we don't. It's just a matter of being efficient and saving energy. Microbes will not waste energy making unneeded proteins. And it's all about competition. Your cousin is regulating herself or himself and saving energy. And if you're going to compete, you need to do the same. Elegant mechanisms to control gene expression. There's gene expression at all of these levels, at the transcriptional level, and then after it's translated into a protein, there's translational control, and then basically everything in between. Okay, we've talked about these a fair amount, these regulatory proteins, and this is now to kind of drill down a little bit. So there's repressor proteins and activator proteins. So we've talked a lot about these promoter sequences upstream of the genes. It's to those promoter sequences that these regulatory proteins bind that either repress, stop that polymerase from synthesizing those genes, or activate the polymerase from synthesizing those genes. A lot of times, in order to exert their effect, they bind to a signal molecule because it's the molecule that is connecting them with the reality of whether they should be expressed or not. So there's that second layer of these ligand molecules, which just means a molecule that binds to a protein. So there's kind of your introduction to activators and repressors. This is still kind of a big picture view, figure 1A, where you've got, sensing internal changes and external changes, and then altering gene expression in response to that. Okay, so here is the gene that needs to be transcribed or not, and then upstream is a separate gene that codes for that regulatory protein. And typically this distance between these two genes is huge. Typically that regulatory protein is synthesized in a completely other part of the chromosome. So if I were you, I would make that, if not a correction, it's just a clarification, because it's typically not in between the promoter and the target gene, is the location of the regulatory gene. Okay, so here is just a big picture view. A ligand binds to a regulatory protein. I'm sorry, I'm seeing these arrows the wrong way. I'm sorry. Here is upstream the gene that codes for the mRNA that synthesizes the protein. Sorry about that. It binds to a ligand and then binds to the regulatory sequence upstream of the gene. So there isn't a lot of information in this slide. It's just big picture showing a regulatory protein binding to a ligand and then binding to the regulatory sequence, which turns on or off the synthesis of these genes right here. Okay, so what's more meaningful is with a little bit more specific. So section B and C of figure 1. Excuse me. B shows you how repressor proteins work. C shows you how activator proteins work. So there's even two ways repressor proteins work. They work by binding in that promoter region upstream of the gene, and when they bind, that represses the synthesis of those genes. And one scenario is this binding of a ligand that once that ligand binds, it stops, it causes the repressor to release from the promoter region, and that allows those target genes to be expressed. And so we call that when the ligand needs to bind to the repressor, and that happens, and the repressor falls off of the promoter and then allows for expression of the genes. We call that an inducer. We call that little molecule an inducer. It induces the expression of those genes because it knocks the repressor off of the repression site. Okay, in another scenario of repression, these are both repressor molecules, the repressor protein has to bind to a ligand in order to then bind to that promoter to repress those genes. So in that case, it's called a co-repressor. That small molecule that binds to that repressor protein is needed to repress those proteins, and so it's called a co-repressor. So the names of those ligands will tell you what that molecule does. An inducer induces the expression by knocking off the repressor. A co-repressor binds to that repressor protein, so now that repressor protein can bind to that promoter. So those are two examples of the regulatory protein being a repressor protein. Okay, then in C, we actually have a regulatory protein that's required for the activation of the RNA polymerase, And so it binds to an inducer, and the inducer now allows it to bind to the operator region. And then this actually promotes the expression of those genes. Okay, so that's described at the bottom in text, 2 types of repression, scenario 1 and 2, versus an activator protein. And we'll look at kind of in specifics a couple of examples of these. Okay, we're interested in, bacteria are interested in, we are interested in sensing changes in the external environment. And so there's a couple times in this chapter where we're going to talk about sensor kinase enzymes, which span the membrane, sensing information on the outside, and then transmitting it into the inside of the cell. And when we say kinase, that means it is involved in phosphorylation events. And as I've said a few times, it is phosphorylation and dephosphorylation, which is a very common way for information to be transmitted. Very simple, and that's where we use this word elegant. It's very simple and is very easy to use and very powerful in terms of communicating specific information. So then the sensor kinase communicates in the cytoplasm with a response regulator, and we've actually looked at a couple, at least in general, an activator regulatory protein or a repressor regulatory protein, which alters transcription rates of multiple genes, typically in bacterial operons. So this is a general feature, or a general thematic or schematic of how the system works, and it's pretty self-evident. Here is an environmental signal, whether it is a toxin or a nutrient that specifically binds to these transmembrane proteins, and it is a kinase enzyme at this end on the cytoplasmic side. So in other words, it's phosphorylated or dephosphorylated, and the phosphorylation event is then sensed by the response regulator. So it becomes dephosphorylated or phosphorylated. It can work both ways. And in this example, it's the phosphorylated regulatory protein that now interacts with the operator region in the promoter region upstream of the gene. And this is a pretty decent slide. When we talk about the operator region, it is the region right upstream of the gene itself that's going to be expressed, and is the point at which those regulatory proteins bind to the DNA. So that's why I say position of the operator is usually in between the promoter and the gene. Okay, pretty straightforward. And we're going to be looking at that in particular when we look at quorum sensing. And with a fair amount of detail, looking at flagellar motility again, taking a longer look at that interesting behavior. Okay, so like I've said, you can control whether that gene is going to be transcribed right at the very first part, the decision to just transcribe it into messenger RNA. You can control it in terms of the stability of that messenger RNA. Sometimes it's immediately degraded. Sometimes it's preserved for a few minutes before it's actually translated. Then if it's translated or not is another level, and then after it's translated, you can control the activity of the enzyme. So like I said, it's a little redundant from my introductory slide 4 or 5 slides ago, but just to reiterate that. Okay, so you've seen some of this information in the LAC operon because it has been used in courses to represent gene regulation for years. It was discovered in mid-60s or early 60s by Jacques Monod and one of the other French fellows that are photographed in your text. And so lactose, understand that lactose is the sugar in milk. And so it's a nutrient. And E. coli has the genes to break lactose down and synthesize amino acids, synthesize carbon, make energy using that lactose milk sugar. And so please understand the big picture. If lactose milk sugar is around, you want to express those genes that degrade lactose and use it as an energy source. If lactose is not around, don't bother making the genes that break it down. So, you know, the rationale for synthesis of the lac operon, you know, start there. Okay, so here's a nice example, and I think we even used this example earlier in the course. If lactose is going to be taken up, it's taken up through a SIM porter with a proton. And that is how that SIM porter works. And so lactose is then taken up into the cell. And now you have a little bit more detail. Lactose is a disaccharide. That the first enzyme that it encounters that is expressed at low levels is beta-galactosidase. And it actually makes it into this allolactose form of lactose. So keep that in mind in terms of that is the actual ligand, that is the actual inducer of the lac operon. So a little bit of lactose gets in the cell, or a little bit is converted to allolactose. Allolactose is that inducer that's going to bind to that regulatory protein. So if you ask the question, where did the allolactose come from, here is the answer to it. Okay, when beta-galactosidase is expressed at high levels, that is the first gene in the operon of the lac operon that degrades lactose. And so you can see it cleaves the disaccharide lactose, beta-galactosidase does, cleaving lactose into glucose and galactose. So that's going to be the first step of the lactose degradation pathway. It's mediated by the beta-galactosidase enzyme. Okay, so here is that enzyme. Beta-galactosidase is the first gene that codes for that enzyme. In the operon, that there are 3 genes shown. So here is a lactose permease, which is this this uptake symport in the membrane. So that's the second component. And then a transacetylase, which we're not going to show that reaction, but it needs to be transacetylated in order for it to be broken down. So like I said, LAC-Z, Y, and A are enzymes or a transport protein, in the case of the permease, that are needed to degrade lactose. And so, like I said, lactose degradation gives carbon and energy to the cell. Okay, so somewhere upstream of the lac operon, the LAC-I repressor protein is made. So, you know, we see this kind of abbreviated messenger RNA is then translated into the actual protein. And then, you know, upstream of those LAC genes, you have the LAC operator in the promoter region upstream of the 3 lactose breakdown genes. Okay, so a couple different scenarios. If you do not have lactose in the environment, don't make the genes, don't make the enzymes that are required to break it down. The substrate's not there, don't express those genes. So we're going to be looking at how those genes are repressed in the absence of lactose. Okay? So here is your regulatory protein, and in this example, it's a repressor protein. It's actually a tetrameric protein, so you can see it's got 4 subunits. And what's really interesting about this regulatory gene is that it actually has 2 binding sites fairly distant from each other, and when it binds to that DNA, it causes that DNA to curl back on itself. And so that's what's showing, and that's pretty unusual. There's a lot of times these regulatory proteins just sit on that operator site and stimulate or inhibit RNA polymerase from synthesizing that gene. But in this example, it actually causes this pretty massive 3-dimensional change, causes that DNA to actually curl up and then around. And so that's what I mean, yanking the DNA so that it loops out. And then if you've got a big wrinkle in the area where RNA polymerase binds, it can't bind. Talk about steric hindrance. This is a massive steric hindrance. When you've got a number of bases curling up, there's just no way that RNA polymerase can get at its binding site. And so this certainly is going to prevent RNA polymerase from transcribing those genes. Okay, so it doesn't really show how, okay. So in the absence of lactose, that's what happens. That repressor protein binds to that operator, and those genes are not transcribed. So, as I've said, in the absence of the substrate lactose, don't express those metabolic genes required for the breakdown of those genes. Okay, scenario 2. How about when lactose, when the cell is exposed to lactose? Okay, let's synthesize the genes. Let's express the genes that are required for their breakdown. Okay, so like I said, beta-galactosidase has a low constitutive level of expression. So there are low levels in the cell all the time. When lactose shows up, and it's just by diffusion, it doesn't have the permease enzyme yet, the symport, it cleaves that, or doesn't cleave, it modifies lactose into this modified form called allolactose. Allolactose is the inducer. Okay, so beta-galactosidase makes allolactose. Allolactose now is shown as these red triangular-shaped ligands, and they bind to that repressor, causing it to fall off. Okay, so if the repressor is repressing, it's bound to that DNA, it falls off, it's no longer repressing. It no longer has that kink in the DNA, and now RNA polymerase is able to bind to the promoter and then synthesize those genes. So remember that allolactose represents the presence of lactose. That is the inducer that binds to the repressor protein, causing it to fall off the DNA, allowing transcription. Now, that's not the whole story. So that is presence and absence of lactose that we've talked about so far. And so like I said, if lactose is present, let's make the enzymes required to break it down so we can gain some carbon and energy from it. So, always return to, does it make sense or not? And it makes sense. They're very efficient, these regulatory systems. Okay. What about other carbon sources? And glucose is the other carbon source that really influences whether those genes are going to be expressed or not. Okay, so in order for that lac operon to be expressed, glucose must be absent. And the reason for that is, and we'll see the data that shows this, glucose is a better carbon source than lactose. There's more energy associated with glucose because you don't have to cleave that disaccharide bond, you just have glucose. And there's more energy associated with glucose than glucose plus lactose. plus galactose. So if glucose is present, that is going to shut down the synthesis of the lac operon. And so let's look at that layer. So in the absence of glucose, cyclic AMP builds up, and this binds to and stimulates an activator protein, which is called cyclic AMP receptor protein. So it's pretty interesting that the lac operon actually has a repressor protein that is involved and an activator protein that's involved. And both are required. The repressor has to be off and not repressing, and the activator